Do you really want to know what ticks me off about you? Maybe the fact that you can't decipher what is and isn't true. No, I didn't sleep with her, and I know I am pretty sure. All I know is I can't stand you.
Alright, we're back with more Scon Biology. I'm your host, Two-Tone Miles. As always, if you're watching this just on YouTube, you should check us out at sconbiology.wordpress.com. If you got questions about the material or just want to talk ska, email me at sconbiology@gmail.com. It's Friday, and the two bands featured in tonight's episode share a common link. They both covered the same song. What song, you might ask? Well, I'll give you a hint. It's Friday. You just heard Duel, the 2010 version by Slightly Askew. They're from Allentown, Pennsylvania, and the song's off the album Striking Out X. And you can also hear the 2000 version on the same album. And it's pretty cool to hear what a difference 10 years makes. The two songs, you can hear the similarities, but there's also a lot of differences. And I like hearing both the old and the new versions of the same song. It's also on their Facebook and Bandcamp pages. And they're working on a new album called It's All for the Kids. And that's going to be due out this spring. And I'll try and give people a heads up when that releases. And on the outro, we've got a song from Fairhaven. So be sure to stick around for that. This is Episode 6, Membranes and Transport. Now, we spent a lot of time discussing the general functions of life. We've defined what life is and the processes that living things do. We've talked about the chemicals essential to life, and we've talked about cells as the fundamental units of life. But we haven't talked in greater detail about how living systems work. And we're getting there, but we still need to do a little more basics. I know, it's a lot of basics. The really exciting stuff is on the horizon. Just keep with me, we'll get there. Now, in the immediate future, we're headed towards studying metabolism. We teased this a little bit on the pizza and beer episode, but this is the basic processes by which living things break down food and convert it to chemical energy. But in order to do that, we need to cover some other concepts. First of all, where does the cell get its food? How does the food get inside the cell? How does it cross the cell membrane? So that's what this episode is about. Now, we talked last time about the cell in the metaphor of a cell as a factory. And we mentioned how the factory needs to be able to import and export products. And this has to be done in an organized fashion. I mean, could you imagine a factory where the day-to-day went something like this? I got a package for you. Well, that's odd. I don't remember ordering anything. Well, I'm going to leave it here anyways. Whatever, man. Is it okay if I just set this here in front of the fire escape? I don't see why it wouldn't be. Now, that factory would be out of business. So when bringing something into the cell, it needs to be done in an ordered fashion. As we discussed in episode 2, life is ordered. It's one of the fundamental things that living things do. Living things are ordered. Now, the cell is defined by its membrane. So this all starts with the membrane. Now remember that membranes are made of lipids, and they repel water. Now, certain molecules can pass directly through the lipid bilayer, just pass right on through. But most can't. And that brings us to the other component of membranes. See, membranes aren't just lipids. Many membranes have transport proteins embedded in the membrane itself that allow molecules to pass in and out of the cell. Now we're going to talk about 4 different ways that things can pass into and out of a cell. Diffusion, the movement of small molecules just by Brownian motion from high concentration to low concentration, and its partner osmosis, the movement of water, also by Brownian motion. Passive transport, where you're moving molecules down a concentration gradient by channel proteins. Active transport, where you're moving molecules against a concentration gradient, and this requires energy. And large particle large particle transport, such as vesicle fusion, pinocytosis, phagocytosis, etc. So we're going to touch on all those, but first, what do we mean by a concentration gradient? Now, a concentration gradient is where you have an area of high concentration next to an area of low concentration. It's that difference of concentration over space that creates the gradient. It can be separated by a physical barrier, such as a membrane, but it doesn't have to be. Now, have you ever had an Italian soda? And I mean a really good Italian soda. Where does all that sugary syrup go? It sinks to the bottom. You take a sip with the straw from the bottom of the glass, it's going to taste really sweet. But if you take a sip from the top of the glass, it'll taste more like plain seltzer water. Now, your glass of Italian soda is a concentration gradient. The sugar is in high concentration at the bottom of the glass, and at lower concentration at the top of the glass. Your Italian soda is essentially a concentration gradient. Now, what do we mean when we say moving up or moving down a concentration gradient? Moving down means going from an area of high concentration to an area of low concentration. Now, let's say you had a box full of styrofoam packing peanuts and a large empty desk. And you take that box and you dump some of the peanuts out onto the desk. That's an example where you'd be moving down the gradient. There were a lot of peanuts in the box. That would be the high concentration. And you spill them onto the empty desk. That would be the area of low concentration. So going from high to low, that's moving down the concentration gradient. Seems easy. Okay, now that those peanuts you spilled, pick everything up, put it back in the box. Now that's moving up the concentration gradient, and this takes more energy. Now it's easy to spill that box of packing peanuts over the desk, but to actually pick everything up and pack it back up, that takes work. Now remember that. Moving down a gradient hardly requires any energy, while moving up the gradient requires quite a bit more energy. Now, because in science we tend to be a little extra confusing, water is kind of a special case. When water moves down a concentration gradient, this is called osmosis. But for everything else, moving down a concentration gradient is called diffusion. Confusing, huh? Now, small molecules can diffuse through the lipid bilayer, meaning they sort of pass through it, but this is a slow process. Think of it like climbing through a hole in a fence. You may need to walk the perimeter of that fence for quite a while before you find a hole big enough to crawl through. The membrane itself is kind of fluid, so there are going to be spaces where things can pass through, but it's not everywhere. And so things are kind of bumping around. Can I go in here? Well, not really. Oh, wait, I can go in here. So that's sort of what's going on here. The molecules are slowly moving through the lipid bilayer, through small pockets of space. Now, you could crawl through the fence, but wouldn't you rather use the gate? Embedded in the membrane are various channel proteins that allow for specific molecules to pass into and out of the cell faster than they would by diffusion, faster and more efficiently. We call this passive transport. The molecules move down a concentration gradient, and this hardly requires any energy. But what if you want to move molecules up the gradient? Like we mentioned earlier, that takes energy. The cell has to spend energy to activate a sort of special gate to move things against a gradient. Since this requires energy, we call this active transport. Now, active transport is going to be really important in some of the future metabolism lessons, as well as lessons on how muscles work and how neurons work. Now, by active transport, you can concentrate charged ions on different sides of the membrane, and this creates a voltage gradient. And not to go into great detail, but without that changing voltage, your brain would basically cease to function. It's those micro changes in voltage that allow our neurons to transmit signals. Now, additionally, active transport can cause a buildup of protons. And we're going to get into this in the metabolism lectures because it basically builds up protons and funnels them through what is basically the biological equivalent of a hydroelectric dam. And it's pretty darn cool. Okay, so diffusion, passive and active transport work for small molecules. But what about the bigger stuff? For the big stuff, the membrane literally folds around an object and engulfs it into the cell, or likewise can expel it from the cell. When cells do this to take in fluid, we call this penocytosis. Think of it as cells drinking. When they take in large food particles, we call this phagocytosis, which can be thought of as cell eating. And when the cell reverses the process to get rid of waste, we call this exocytosis. Now we got a few more things to talk about here. Maintaining the proper concentration gradient is hugely important if you're a cell. Now if you take a cell and put it in a highly concentrated salt solution, the water is going to quickly leave the cell, and the cell's going to shrivel. Now if you go the other way and put the cell in basically a pure water or low concentration solution, the water's going to flow back in and the cell will swell up. It could even burst. Now that's why this idea of blood sugar is so important. Now your blood needs to have the right sugar concentration to feed the body without overloading it. Too little sugar and the cells are not going to be getting enough food. They could die. That's a bad thing. But if you have too much sugar in the bloodstream, the cells are at risk of bursting, and that's also a bad thing. Now blood sugar needs to be kept in an optimal range to stay healthy. You need to keep your blood sugar not too high, not too low, in the middle where it's just right. Now lastly, I want to add in that channel proteins for active and passive transport are not the only proteins you find in the membrane. In the membrane, you may also find enzymes, and we're really going to hit on enzymes hard in the next lecture. You can also find signal proteins. These are proteins which respond to signals from the environment and tell the cell to react to a signal. You have cell-to-cell recognition proteins, and these proteins basically help identify what the adjacent cell is in a multicellular organism. You can have joining proteins, proteins which literally fix the cells together, hold the cells together into tissues. And you can have proteins which bind part of the cytoskeleton. And we covered cytoskeleton in Lecture 5. That may have been on the WordPress site, so I think we covered cytoskeleton in Lecture 5. If not, shoot me an email. Again, sconbiology@gmail.com. Okay, so that's going to be enough membranes for now. Now, playing us out is Upside Down by Fairhaven. And I was going to use the track Here to Stay, but they talked me out of it. So we're going with Upside Down. Now, if you want a little extra credit, go out and check out Here to Stay. And while you're at it, go check out all their songs. They're out of Davenport, Iowa, and they're young enough to still be in a high school biology class. They opened for the Toasters on their 30-year anniversary tour, so a big tip of my hat to you guys. They don't have music for sale just yet, but they got their music out there on Facebook. You can also find it on YouTube under the channel Fairhaven TV. And we here at Ska and Biology want to show big support to these young guns of Navarone. So here's some Fairhaven for you.
Well, I'm pretty sure this isn't right. If you think about it, the way she treats him, can't do nothing about it. She came off as the perfect girl, had everything in common. Now she's really mad, but that's not uncommon. Maybe if I grew up, she would like me, make things like they used